Okay, I'm back with an informational screencast having to do with multiple alleles. All right, so you've watched the Amoeba sisters, and you know what? Walk, I should say run, don't walk, to subscribe to their YouTube page. They are great. They also have a website, amoebasisters.com, handout for each screencast. It's a great way, and I'm going to use some of theirs going forward because they're a lot more polished than your teacher, who's a much more upfront person than they are. I just wanted to give you some pointers here of things you may see, but the main content is coming from them. All right, so we go back to the smartboard for a couple of minutes here. All right, I wanted to point out to you that multiple alleles also exhibits two things: complete dominance, which I'll touch on at the end, and codominance. And blood type AB is codominant type 1 because AB is considered to be a new phenotype. It's not A and B, it's blood type AB. So often enough questions will blend complete dominance with codominance, okay? Keep that in mind. Keep in mind also the beginning of the screencast where they explained very well how to tell and how you get a blood type, all right, which is not quite genetics, but it does give you a little bit more of the circulatory system. So for example, if you don't have any antigen on the surface of your red blood cell, remember the antigen is nothing more than a cell surface glycoprotein. That means antigen A. If you have that, your blood type A, you have antibody B, so forth and so forth. Now, blood type O, again, remember she highlighted because blood type O can only receive blood type O. All right, blood type O has both antibodies, and therefore, if the blood type O person, like myself, received any other blood type, the blood would start clotting, as you'll see on the next slide. It's called agglutinizing. All right, so you want to be very careful with blood types and who gets blood types there. All right, going to the next panel, you see here a fun lab that seniors do sometimes. I don't take the blood from freshmen. But you can give if you have to find out which blood type you have, and they colorize, you can give anti-serum A, in other words, antibody B in one and antibody A in the other. So it's anti to blood type B. So notice what happens if you were giving anti-serum A, which contains the antibody B in it, bam. And that's the way you tell, because if your blood clots with antibody B, you've got blood type A, blood type B, AB, and nothing with blood type O. It's worth noting that genetically, blood type O is the most wide blood type in the gene pool. All right, about 47% of the population has one form of blood type O, O positive or O negative. All right, let's go to one more panel here. All right, and that's how you can show the blood type. You saw that she used the classic immunoglobulin line. All right, so you'll see questions that look like this, but this II looks the exact same genotype as AA. And we do not care, meaning me, how you show it, whether you use the I's or you use the capital letters. There are 6 overall blood type genotypes: AO, AA, BB, BO, AB, and OO. OO is equal to the II, the small I. So blood types do show complete dominance. AO, BO, and OO, or blood type A, B, and O in that case, show complete dominance, meaning blood type A or the A genotype is complete over, all right, O. Dominant over O, dominant over O. So here is your homozygous recessive, homozygous dominant, and 3 heterozygote. There, all right, true heterozygote, codominant AB. And one other thing that you'll look up, she discussed at the end, was what it's called hemolytic disease. Basically, what would happen if a pregnant woman had either a different blood type than her baby or a different Rh factor? Is that good? Is that bad? You're going to do a little research on hemolytic disease. So chapter 23 in your book, I think this is 23.3. A little bit on multiple alleles from me. Watch the Amoeba sisters.